Hi guys, this is a big day. We finally survived our photosynthesis and cell respiration, all of our um, metabolic reaction chapters, and so now we're finally on to some of the better stuff. So we get to talk about, in the next few chapters, we'll be talking about mitosis, we'll talk about meiosis, and then we'll also talk about a little about some reproduction. So essentially, um, cells of all types can grow and divide. We talk initially about prokaryotes first because they divide a little bit differently. You get more prokaryotes by a process called binary fission. And so essentially that's literally just splitting in two. So it's a very simplistic process that involves the production of two daughter cells that were identical to the original cell. And so when that happens, you will just have a simplistic like ring of DNA that has a few proteins, and that's the chromosome. And then it is replicated into two rings before division, and it attaches to the plasma membrane and it simply divides. So there's no spindle involved in this process. It's very simplistic. It's literally just bam, 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 and then you have two identical cells from this process. For eukaryotes, though, it's a little bit more complicated. Eukaryotes actually have to go through eukaryotic cell division. And so for eukaryotes that reproduce asexually, mitosis is the way that happens. Eukaryotes who reproduce sexually utilize meiosis as a process. And so mitosis itself involves a cell dividing itself and the DNA to form two identical daughter cells, whereas meiosis is very different because the cell will undergo two rounds of nuclear division, and you're able to produce at the end of that process 4 gametes. And those 4 gametes are not genetically identical, and they're haploids. They have half the genetic information that they started out with, so that makes it a little bit unique. So prokaryotes and eukaryotes actually divide just a little bit differently, right? So both binary fission and mitosis allow daughter cells that are they create daughter cells that are genetically identical to the parent. So when you're single-celled, that's really easy. Binary fission is just boom, boom, just dividing up and very simplistic. When you're multi-celled, it becomes a little bit more complex, and that's why we use mitosis for growth, renewal, and repair. And that happens in fungi, plants, and animals. So consider, like anytime you get a boo-boo, mitosis is going to create 2 genetically identical daughter cells, or more, right, depending on your growth signals at that point or whatever needs to be repaired. You're going to have the same cell be created to fix that. So this comes from your textbook, and it just shows you the type of organism, how they divide cellularly, and what they're using it for. So like bacteria and archaeans use binary fission for asexual reproduction. There are protists and some fungi that actually use mitosis for asexual reproduction, and then everybody else, fungi, plants, animals, they utilize mitosis for development, growth, and repair only, so not sexual reproduction. So the cell cycle as a whole is a set of stages from the first division to the time resulting in daughter cells dividing. And so there are 2 major stages of the cell cycle. The first is interphase, and that interphase gets divided up, and the cell actually remains in interphase for the biggest portion of time. And then comes the mitotic stage, and that includes mitosis and cytokinesis. And the cell spends just a smaller amount of time undergoing mitosis. So just before the next division, when there is a signal that division needs to happen, um, the cell will grow larger, the number of organelles will double, and then DNA actually gets replicated. So this shows you the cell cycle as a whole. And so what you can see is that if you look, the bigger portion of the cell cycle is spent in interphase. And so then just a tiny bit of this is actually spent in mitosis. So interphase does involve some important things, because essentially, when you think about it, interphase is just preparing the whole entire cell to divide up its nucleus and its contents. So mitosis is nuclear division. And if you think about what's inside of a nucleus, we learned about a cell, and when we think about what's inside of a nucleus, like DNA, the creature's whole entire being is defined by that DNA. And so when you're dividing the thing that contains the DNA, you want to do it pretty orderly, and you want to make sure everything is right. So your DNA gets split and divided equally and sent to the daughter cells correctly so that you don't have mutations, because it's your DNA. Like if there's any kind of mutations, that's gonna change your whole entire cellular makeup. You don't want that. So it's a very orderly process. And there are checkpoints along the way, if you look, that ensures this process is done well. It's not willy-nilly. Like cells aren't just dividing willy-nilly without any regulation. So most of the cell cycle is spent in interphase. I said that. And during this phase, for the most part, the cell is performing its usual functions. And of course, it depends on what kind of cell you're looking at with regards to how long the cell spends in interphase. Like, I mean, if you're talking about embryonic cells, we're like bam, 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 multiplying very rapidly. And it's divided up into 3 stages. So there's the G1, there's the S, and the G2 stage. And note, nerve and muscle cells don't complete the cell cycle. They remain in an arrested G0 stage. So if you ruin a nerve or muscle cell, eek. So the G1 phase is called the gap phase, and this is where you'll have recovery from the previous division. The cell will double its organelles, so we need 2 of everything. If we're taking this one cell and we're dividing it up and we're making 2 genetically identical daughter cells, we're gonna need double the organelles. And it also prepares for DNA synthesis. Then in the S, or the synthesis phase, that's where you'll see DNA replication, and you want to do this very well. So chromosomes enter with 1 chromatid each, and they leave with 2 identical chromatids called sister chromatids. And then the G2 phase is kind of between DNA replication and then before mitosis starts. So here's a chromosome. What you'll see is it has been duplicated, and if you look, you've got the yellow guy, and then the blue guy is just a replicate of the yellow guy, and so that's why they're sisters. So the mitotic stage is the more complicated stage. So this is where you're going to see nuclear division. So this is where you're seeing daughter chromosomes that get distributed by the mitotic spindle. We've heard about the mitotic spindle. The spindle is not a stranger to us. Way back in the day when we were talking about the organelles of a cell, we talked about those cytoskeletal elements, and we said that there were some big cytoskeletal elements that actually had a big job. This is when those come into play. So we'll see those, we'll see the nucleus divided, the contents of the nucleus divided and separated into two daughter cells. And then finally, once they're ready to split completely apart, we'll see cytoplasm. And that results in two genetically identical daughter cells. Remember that. So the whole cell cycle as a whole is controlled by internal and external signals. Like, it's not like, you know what, I'm bored, I think I'm just gonna mitosis today. That's really not how it works. So essentially, it's controlled by signals, and remember, the cells don't get text messages, and they don't get, emails. They don't have an Insta or anything like that. No fans only. What they do have are signaling, and so those are chemical signals. When cells are communicating, it's a chemical signal. So internally, the signals are a little bit different than externally. Externally, you'll see growth factors like signaling proteins, but internally you have these cyclins that are proteins that increase and decrease as the cell cycle continues. And when those cyclins drop, that's where you see the cell cycle stop at G1, M, or G2. At those checkpoints are designed to say, hey, check, everything looking good, everything going well, our organelles doubled, our DNA replicated, everything looking good. And if not, at those checkpoints, the cells will be like, it's not looking good, guys, and then it'll attempt a repair. If a repair isn't made, that cell will take itself out. You will not see the continuation of the cell cycle if everything didn't go right. Otherwise, you're going to be doubling a bad cell that's mutated, which we'll talk about that when the cell cycle goes rogue. So when we stop at a checkpoint and like the cell will notice that nothing looks right, something happened. If something were to, if we see that happening, if the cell notices that, or there's signals that show that happened, then what happens is the cell will undergo a process called apoptosis. And apoptosis is programmed cell death. So it's almost like in those old spy movies when the spies would carry around like a little capsule. If they were caught by their enemies, they would take it and just take themselves out. Cells have the same thing, and it's not unnatural. It's literally a way to manage cells. So it's programmed cell death. It's gonna happen. It's essentially the process. Like, your cells just don't go around taking themselves out because it's held in place by caspases that are stored in the cell and inhibited until it's needed. And so apoptosis results in the nucleus fragmenting, the plasma membrane blisters, cell fragments are engulfed, and that takes that cell out of the game. This shows you what it looks like. Apoptosis is really good for taking out mutated cells, but it's also kind of a required thing. Like in fetal development, if apoptosis didn't have it, we would all still have webbing between our fingers. It helps destroy infected, abnormal, or cancerous cells. For those individuals who go through metamorphosis, it's what helps break down and rebuild the entire structure of an organism. So before we talk about the division of the nucleus, or mitosis, we need to kind of understand that DNA is actually found in very long threads. And so it's stretched and intertwined between divisions, and it's kind of associated with these proteins called histones. When DNA and histones are intertwined around one another, they're collectively referred to as chromatin. And so when we are looking at mitosis and we're studying mitosis, what we'll notice is that the chromatin condenses and it coils into very distinct visible chromosomes. So when we see chromosomes, we're only able to see them because they're condensed and coiled and associated with those histones. And then they become those visible, and they kind of become those distinct like X patterns that we're used to seeing with chromosomes. And each species has a different characteristic chromosome number. And I want to remind you that chromosome number does not necessarily indicate the complexity of the organism. So this shows you just an image of DNA associating with those histone proteins and the becoming a very visible condensed chromosome. So this is a chart that comes from your textbook that shows you the chromosome number of different organisms. And just so you know, the chromosome number of a let's see, the chromosome number of different organisms is very unique. Look at the chromosome number of the fern Ophioglossum vulgatum, 1,320. That's quite a bit different than our chromosome number, for sure. So, we get half of our chromosomes from each of our parents. That is referring to ploidy. So it's a number of complete sets of chromosomes. So the diploid number, which is always indicated with a 2n, indicates that there are 2 sets of chromosomes of each type. The haploid number is showing 1 set of chromosomes. So humans have 2 sets, one from mom, one from dad. Humans have 23 different types of chromosomes, but each of those types is represented twice in each body cell. No, I said body cell only, and no, I said twice. So you got half of that number from your dad, you got half of that number from your mom. You get 23 chromosomes from dad, 23 chromosomes from mom, and they join together. So the diploid number that you get as a human is 46 chromosomes. Note that gametes are haploid. Like when we make our gametes in our bodies, we do that using meiosis, and we're going for the creation of, our goal is to make haploid gametes, because when we join those gametes together, we want to make a diploid organism with the correct number of chromosomes. We'll be making no fern. So at the end of S phase, each chromosome is internally duplicated. So you'll have 2 identical DNA chains, or sister chromatids, and they're attached at a single point called a centromere. And so when I imagine sister chromatids attached by a centromere, I really imagine like hair held together by a hair tie. I don't know. So during mitosis, those centromeres hold those sister chromatids together. Eventually they do separate and they get distributed to the daughter. Each becomes a daughter chromosome and they get distributed to the daughter cells. So here's the sister chromatid. Here's, if you look here, here's our centromere. There are also these weird things called kinetochores. This guy right here, the yellow thing. The kinetochores are like protein attachment points for the microtubules that are actually going to kind of push and pull these chromosomes around in the cell so that eventually they'll line up so that they can separate correctly. These are like tow hooks for your vehicle you're taking out to bricks. So just outside the nucleus is the centrosome. We've heard about the centrosome, we talked about it a long time ago, but it's the microtubule organizing center in animal cells. Note I said animal cells only. Plants don't have to have a centrosome because they don't have to worry about organizing the microtubules so much because of the way that they go through cytokinesis. It's so different. So the centrosome organizes the mitotic spindle. So the spindle is the thing that's going to send out those microtubules so that they can attach to those sister chromatids and move them around the cell. So we also replicated one of those in S phase. So now we have two before mitosis begins. And so they're oriented at right angles to each other. And they're 9 triplets of microtubules arranged in a cylinder. And so they will separate and they will create the plane of division for a cell. So if you look, they look like little delicious churros. So the phases of mitosis include prophase, metaphase, anaphase, telophase, and then cytokinesis. So during prophase, the chromatin gets condensed, it's distinguishable. You can see that little X dude. The nucleolus will disappear, the nuclear envelope disintegrates, the spindle begins assembling. Those centrosomes move away from each other, and you're going to start seeing like microtubules form. During prometaphase, the centromere of each chromosome develops 2 kinetochores. So those kinetochores are the things I showed you in the previous slides that attach to each sister chromatid, and they almost are like little tow hook attachment points where the microtubules will connect and push and pull those sister chromatids around the cell until they're ready to divide and go to the daughter cells. Metaphase is one of my favorites because it's easy to distinguish. During this phase, all of the sister chromatids line up across the equatorial plate or the metaphase plate, and that's where the mother cell will be divided. In anaphase, the centromere dissolves, releasing the sister chromatids, and then they separate, and now they're called daughter chromosomes. So once they separate, they become daughter chromosomes, and they are pulled to opposite poles along kinetochore fibers. During telophase, the spindle disappears. You'll find 2 clusters of daughter chromosomes. The nuclear envelopes will start forming around the 2 baby daughter nuclei, and each daughter nucleus receives 1 chromosome of each type. During cytokinesis, this is literally, the term means dividing that cytoplasm. And so that divides the mother cell's cytoplasm equally to the daughter nucleus. It encloses each daughter cell in its own plasma membrane. And what happens is a cleavage furrow will begin to form between those cells, right? And it eventually, you'll have these like little tiny fibers that will help pinch the mother cell into, almost like you're pulling your drawstring of your hoodie, and that pinches that mother cell into. So that's what it looks like. You'll have a contractile ring that will get tighter and tighter and tighter until it pinches. Plants are kind of unique though, because plants unfortunately have that cell wall, and that does not allow that cell to just be pinched in two, right? So for cytokinesis in plant cells, a cell plate has to be created. So that is essentially where these little membrane-bound vesicles form right in the center of that cell at that plane of division, and then the daughter cells will secrete primary cell walls on either side of that cell plate. It becomes filled with middle lamella, so you essentially build a wall that separates that plant cell, and then you'll have individual daughter cells. This is an image from that shows all the phases in animal and plant cell mitosis. It shows every single one of them. You can find this in your textbook. You can find this in your lab manual. It's great study material. We'll look at them in lab very closely as well. So this is a representative of early prophase, where the centrosomes have duplicated, chromosome is condensing, nuclear envelopes going away. For real prophase, no more nucleolus, duplicated chromosomes are visible, centrosomes begin moving apart, you can start seeing the spindle. In prometaphase, the kinetochore is attached to a spindle fiber, Those polar spindle fibers stretch from each spindle pole. During metaphase, the centromeres of chromosomes are then aligned at the metaphase plate. You can see that. During anaphase, the sister chromatids pull apart. They become daughter chromosomes at this point. The centromeres are dissolved. Each pole receives the same number and kinds of chromosomes as the parent cell. You don't want any mutations. During telophase, the daughter cells are forming as nuclear envelopes and nucleoli reappear. Chromosomes become indistinct again. So the whole purpose of mitosis is for growth and repair. And for flowering plants, the meristem tissue can divide all through the life of the plant. But for mitosis, like for mammals, mitosis is that thing that allows us to correct boo-boos when we get hurt. When a fertilized egg becomes an embryo, mitosis happens in great amount. When that embryo becomes a fetus, lots and lots of growth, lots of mitosis. When a child becomes an adult, if you get a boo-boo, if you break a bone, mitosis is going to allow those cells to replicate and create more cells to fix it. Now, there are some mammals do have stem cells. So stem cells are unique because they retain the ability to divide. So like red bone marrow stem cells divide to produce various types of blood cells, and they continue doing so. It's only controversial to talk about stem cells. Like, I think it's just the location of where you're getting the stem cells. It is a hot button topic in a lot of ways. Because stem cells they have the ability to divide forever and ever, like we don't. Our growth pattern, we go through mitosis until we become an adult, and then we get in an arrested state. We're not growing anymore. Like, I keep waiting to get taller than 5'3", but it's not going to happen. So now if we get hurt, our body's going to fix that. It's not like, oh, this is the end. I'm sorry, you're grown. You no longer get to repair yourself. That's not the case. But we have a growth pattern, and that's where we'll stop at. Now, stem cells don't have that limitation. Stem cells can divide and divide and divide, and that's why, of course, scientists decided to use them for research. And so we know we can use stem cells to produce human tissues using the originator's same cells, and that's called therapeutic cloning. And that's important because, like, if you need an organ and you don't want to, like, you want your own cells so that way you don't suffer through rejection, that's a great idea. Now, when embryonic cells are used for reproductive cloning, that gets crazy. So I don't know if you've ever looked into Dolly the sheep that was created from cloning, but it happened. I don't think Dolly had a great quality of life, but it was a thing that happened. So this shows you the two different types of cloning. And most people aren't so opposed to therapeutic cloning because they're like, I need a liver. Can I have a spare? Oh, it would be great if I could grow my own. Yay. But when it comes to cloning a whole individual, that's when things get kind of sketchy. Okay. So we talked about the normal cell cycle. We talked about mitosis, how the whole process works, how it's very, very organized. But sometimes cells still go rogue, and that might happen as a result of some genetic predisposition that it's going to go rogue, or it could be some external mutation causing that cell cycle to go rogue. And when that happens, that's when you will see abnormal growth of cells, and that's called a tumor. So benign tumors are not cancerous. They're encapsulated. They don't invade neighboring tissues. They don't spread. Not to say that a benign tumor can't cause issues, because it can. Like if it's in the wrong spot, it can take up space where your organs need to be. but it's not gonna spread like a malignant tumor. Malignant tumors are cancerous. They're not encapsulated. They readily invade neighboring tissues. They can detach and lodge in distant places called metastasis. And we usually see this as a result of the mutation of genes that regulate the cell cycle. So cancer cells are kind of unique because they usually have some certain characteristics, some common characteristics. First of all, they lack differentiation. So like your kidney cell, it looks like your kidney cell and it is always going to be your kidney cell. Cancer cells and those kidney cells, they're actually helping to assist in the function of that kidney. Cancer cells are just cells and they don't look like the cells they're supposed to be doing, and they're deadbeats. They're not really helping the organ or the location out at all. They're usually pretty identifiable by having abnormal nuclei. They don't undergo apoptosis, and that's the trick here. Like usually you're like, oh hey, but we have apoptosis, we can get rid of those bad cells. They're not susceptible to apoptosis. They're likely to form tumors. And so normally what happens when your cell gets a growth signal, mitosis happens, great, let's build another cell, okay. Then your cells kind of bump into each other and they stop. They have this signal like, okay, I've touched another cell, I know I shouldn't keep growing. Cancer cells don't have that signaling. They don't have that ability. So they'll keep going through, they'll keep dividing and dividing and dividing and making more cells until a tumor forms. And typically that tumor will break off or metastasize and end up in a whole completely different part of the body. Then what's even worse about, okay, you're like, oh, well, okay, that cell broke off of the body, broke off the tumor. It doesn't have access to oxygen or important resources, so it's not going to make it. No, but it can. It's so evil. In that, what happens is wherever they lodge, they can form their own blood vessels and get their own nutrients anyway. So they're so susceptible and so successful, and that's why cancer can be so tricky to treat and so tricky to get under control, is because of all of these things, these critical things that cancer cells are able to do. So this shows you the progression of cancer, how you'll form a tumor. If you look at this spot right here, this is kind of critical because you'll see that tumor can end up metastasizing, and if that tumor, those cancer cells metastasize and spread to the blood vessels or the lymphatic vessels, those are basically superhighways. Both of those systems are superhighways that can end up carrying those cancer cells all throughout the body. So you may have started out with prostate cancer, or you may have started out with testicular cancer, but later on down the road, you may find out that it had already spread to your lungs and you had no idea. That's how that happens. So this kind of just shows the difference between cancer cells versus normal cells. So cancer cells have non-differentiated cells, where normal cells do have differentiated cells. They're kidney cells and they're doing kidney work. That's what they're doing. Cancer cells have abnormal nuclei, where normal cells have normal nuclei. Cancer cells do not go through apoptosis and normal cells do. Cancer cells don't have contact inhibition. Remember, like they're going to form a tumor because they touch each other and they don't care. They're going to keep going. Normal cells have contact inhibition, where they touch other cells and they're like, oh, we're done with mitosis, guys. Let's stop. Cancer cells are disorganized and multi-layered. Normal cells have one organized layer. Cancer cells undergo metastasis, where normal cells remain in the original tissue where they belong. Essentially, the origin of cancer cells is generally because genes get mutated. So two types of genes may be mutated when there's an imbalance or an abnormal balance. Oncogenes or tumor suppressor genes. If either of those genes get mutated, cancer cells can form. There are other causes of cancer as well. So chromosomes typically have special material at the end called telomeres, and they get shorter after every cell division. And when they get really short, the cell will no longer divide. Now, there's an enzyme that regulates the length of telomeres, but if there's a mutation in that telomere gene, in the telomerase gene, the enzyme that regulates telomere length, cancer cells can form.